Hi all, so this is going to be the review of my Chapter 10 notes. Hopefully you don't hear the minpin who's going crazy in the background. But this, remember for the full Word document, look under the Week 2 questions thread and the My Notes post. Okay, so this is going to be kind of DNA based for this chapter. So in the 1800s, we know DNA is in cells, but we do not understand the role of heredity. By 1930s, we knew that chromosomes do have genes. We think chromosomes might have to do with inheritance. By the 1940s, chromosomes are made of protein and DNA. They're in love with protein, though, as scientists. They just think it does everything, because it does. But it is that kind of derails people, and it takes until the 50s to fully realize the role of DNA. Molecular biology is the study of heredity at the molecular level. DNA, as you remember from a previous chapter, has 4 bases: T, C, A, and G. RNA changes the T to a U. Otherwise, it still pairs with A, C with G. Let's scroll down. There's a checkpoint with GG dot. In 1962, Watson, Crick, and Wilkins received the Nobel Prize for their work. Franklin, who I believe deserved the prize as well, but she died of cancer in 1958 and did not receive it. At this time, Nobel Prizes could not be awarded to anyone who had already passed on. Watson was the professor at UCSD when I went there and drove a car with a license plate that said ATTC. So, just a little fun history. Here's some songs that might help you. Well, a song that will help you with the cells, and then a little bit on DNA replication by PBS. Excuse me. PBS seems to be a really good source for some videos. Good explanations without dumbing it down too much, right? Let's go over this checkpoint. What's transcription and translation? Transcription is the transfer of genetic information from DNA to RNA, so it's going to be making a copy of it. Translation is to use that information from RNA molecule to the synthesis of a polypeptide. So transcription, you're just copying it. Translation, you're turning it into something else. Let's scroll down. And here's a good picture of it. So for transcription, we're at the RNA. Then we go outside of the nucleus with that RNA, and translation leads into our protein. Remember, we love proteins because they do everything. Here are some fun videos. Transcription, like I said, synthesis of RNA by DNA. Translation uses mRNA, messenger, because it moves around, to make protein, which is our polypeptide. Some fun videos. Good protein synthesis rap that's amusing. So in the 1940s, from the work of the American geneticists George Beadle and Edward Tatum, with bread mold, Neospora crassa, Beadle and Tatum studied strains of the mold that were unable to grow on the usual growth medium. Each of these strains turned out to lack an enzyme in the metabolic pathway that synthesized an amino acid called arginine. Beadle and Tatum also showed that each mutation was defective in a single gene. Accordingly, they hypothesized that the function of an individual gene is to dictate the production of a specific enzyme. Now that means the function of a gene is to dictate the production of a polypeptide. Let's go down. So one DNA codon is going to be 3 nucleotides. This leads to one RNA codon, which is 3 nucleotides, and that leads to 1 amino acid. So it takes 3 letters to end up with 1 amino acid. Make sense? Genetic code, the set of rules relating nucleotide sequence to amino acid. So you have those 4 letters, and those are going to create all the various amino acids, which we'll have a chart really soon. Let's see. Is there a chart? There's a chart. So here's all the various codons. Let's go to 100% so it's easier to see. See, so all of these codons, these are the letters that need to be on our mRNA, which is translated from our DNA to create the codon. And some of them stop the process, but only one of them starts the process. Okay? And then depending on what you have is what your protein looks like. I can make it nice and big because I'm old and I like to see. Let me pause and yell at the minpin. Okay, hopefully my miniature pincher will be quiet and we can continue. So let's scroll down. Another checkpoint. We already talked about that. Alright, so here we have another. So the initiation of transcription. This is going to be really, really specific. Basically, you just need to know the whole amino acid leads to protein kind of thing, with the mRNA giving you what the amino acids are going to be. But I'm going to get into the specifics just to help you understand a little better. So to initiate transcription, the start transcribing signal is a nucleotide sequence called a promoter. So here it's green and labeled promoter in case you're colorblind, which is located in the DNA at the beginning of the gene. So before you have the gene, you're going to have a promoter. Makes sense. The first phase of transcription, called initiation, is the attachment of the RNA polymerase, that's our little polymerase, to the promoter and the start of RNA. So you see here he's going to stick, and then elongation, he goes through and he goes copy, 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 copy. Gets to the terminator DNA, it goes, ha, no more. And then we have our ribbon of mRNA. See if we So messenger RNA, mRNA, the messenger molecule that is translated. So it was from the DNA, we created an RNA, and we got our mRNA. That's our mRNA. Transcription is similar to replication, except makes mRNA that cannot leave the nucleus. Transcription factors bind to the promoter. RNA polymerase separates the two strands and joins the RNA nucleotides to DNA at the promoter, and going from 5'3", shocking, I know that's what it always does, and ends in the terminator. Okay? So are you copying the DNA to make another DNA, or are you copying the DNA to make just RNA? It's a big difference. Okay, so after transcription, we're going to have to process that RNA. It goes, the 5' end goes in, it gets a cap, which is modified with the G, gets a poly A tail, which is a bunch of A's on the end, the 3' end. RNA splicing by introns is done by the spliceosome, which also binds the exons. What this means is we're going to stick a little top on it, or stick a tail on it so it can wiggle around, and we're going to get rid of anything that's not coding and just leave the stuff that is coding. Oops, that was way too far. There we go. Scroll down a little bit. Yeah, and it says what the introns say, cut me out. Ah, good enough. So let's see what this is going to look like. So here's our mRNA. Here is the next amino acids goes here. It's going to plunk over to the first doohickey, and then we code the next one. We just keep the chain growing. So we're going to keep the chain growing. And there's a good little video. So translation, let's talk about that. That's what we're doing here with our polypeptide chain, right? mRNA goes from the translation site of the ribosome complex, shockingly red, 5' to 3'. You see the tendency, we like to go 5 to 3. mRNA is attached to the small ribosome. The initiator tRNA attaches, and the large ribosome attaches to mRNA. It looks like a baseball mitt. Then we got the amino acid tRNA synthase, verifies the amino acid on the tRNA, and the tRNA is processed through the large ribosome according to their anticodon. The third base is called a wobble because it's not so important. So it just means that tRNA matches up with the mRNA. If you get a chance, watch this video because it is hilarious. Hilarious. Little slow in the beginning and then it goes crazy. Alright, let's talk about some mutations. which I think is the fun part. Mutations within a gene can occur as a result of base substitutions, deletions, or insertions, and a few other things, but those are the ones we're going to focus on right now. So base substitution is where an A replaces a G in the fourth codon of the RNA. The fourth. So the result is a polypeptide. It should be the third codon, right? We're always dealing in thirds. And this is where the polypeptide is a serine instead of a glycine. The amino acid substitution may or may not affect the protein's function. Sometimes, if you notice, like I said, that third codon, it's not too picky, so it could just end up in the same thing, or it could change it. A nucleotide deletion is when the nucleotide is deleted. This can cause all sorts of issue, because now all the codons that are after the deletion site get moved up one, and they're all going to be misread. This is probably going to lead to a non-functional protein. And then we have nucleotide insertion. This is similar to deletion, just the opposite. Now we're sticking in a new nucleotide, so everything after the additional new nucleotide is going to be different than what it was originally meant to be, right? Also likely to lead to a non-functional polypeptide. So why do mutations occur? Environmental reasons, genetic reasons. Mutations resulting from errors during DNA replication or recombination are known as spontaneous mutations, as are other mutations of unknown cause. Other sources of mutation are physical and chemical agents called mutagens. The most common physical mutagen is high-energy radiation, such as X-rays and UV light. Chemical mutagens are of various types. One type, for example, consists of chemicals that are similar to normal DNA bases, but that base pair incorrectly when incorporated into DNA. Many mutagens can act as carcinogens, agents that cause cancer. Mutations are one source of a rich diversity of genes in the living world, a diversity that makes evolution by natural selection possible. So, yes, many mutations are bad, but sometimes mutations can be good. That's how all of life evolved. They finally got a mutation that did something useful. Blah, blah, blah. Checkpoint. Go through it. Let's talk about viruses. A virus is nothing more than genes in a box. It's a bit like a nucleic acid wrapped in a protein coat. A virus can survive only through infecting a living cell with genetic material that directs the cell's molecular machinery to make more viruses. There used to be a whole debate on if they're alive or not. There's a good video to watch. Viruses that attacked bacteria are called bacteriophages, bacteria eaters, or phages in short. I have a minpin who's just barking. Lytic cycle. After many copies of the phage are produced within the bacterial cell, the bacterium lyses or breaks open. So basically it's just so full of these viruses that it kind of explodes. The lysogenic cycle is the viral DNA replication occurs without phage production or the death of the cell, so it can actually survive it. Which do you think is actually better for the virus ultimately? Is it better to be more a mutualist or a parasite? I guess we'll talk about parasites later. I think that's week 4. Is that everything? No. No chapter should be that short. Okay, so then here's an example. See, lytic cycle, bacteria goes explodo. Lysogenic cycle, it's all good. So to infect a plant or anything, a virus must first get past the plant's outer protective layer of cells. For this reason, a plant damaged by wind, chilling, injury, or insects is more susceptible to an infection than a healthy plant. Makes sense. How much is left here? Let me check really quick. Yeah, let's pause with this plant and we'll continue on in part 2 because I only have a few more seconds before my video will say, haha, you're out of time. So check out the next video to see the rest of chapter 10.